Hey there, beautiful people. It's the Shower Guy, and you're tuned into Lauren and PJ Do Science. Today's episode, mRNA, Part 1. Galileo, Newton's laws, Darwin writes on nature flaws, Curie, radium, and X-ray beams, Einstein relativity, Watson, Crick, and DNA, black holes, Hawking, math, Copernicus redraws a path. Lauren and PJ do science. She's got a doctorate, he's got a beat in lyrics. Lauren and PJ do science. Yeah, he's a broke musician, and she's gonna teach him. And now your hosts, Lauren and PJ. All right, yeah, thanks, Shower Guy. Yeah, like you said, we're going to be going over mRNA today and learning all about that in the next couple episodes. But before we get started, Lauren, how are you doing on this very fine day?
I'm doing fantastic, PJ. Thank you.
Hooray! I'm surviving. I'm surviving. I'm getting a bit of that winter ick in me. So like that allergy I get every year. It's gross.
I kind of feel it like on the periphery, like it's waiting in the wings to get me. And it's like, okay, once this semester is over, I'm going to become horrifically ill. But until that happens, my body is just refusing to allow it. So,
yeah, I have the, it feels like somebody has taken like, what's his face? The Iron Claw, wrestling. Oh my God. Von Erich. Which one was it? Dude, any day of the week I could spit this out except for now. But one of the Von Erichs had like the Iron Claw, and it feels like he's doing that to my face right now. So
oh, I'm so sorry.
I'm so embarrassed because I'm such a wrestling fan. You think I could spit that name off the top of my head, but I'm completely blanking.
You could
right
tell
now.
me any name, and I'd be like, yeah, just like that guy. Like, I have no idea. So
well, here's the thing: there was like 27 Von Erich brothers, so I'd have a 1 in 27 shot of getting it on the first try. It might have been Carrie. I don't know. Somebody in the comments correct me, or I could just look it up after the show. I don't know. Just don't at me wrestling Twitter and podcast and whatever, just because I know who it is. I'm just completely blanking right now. But anyway, yeah, that's the kind of shape I'm in. I just feel like my face is being crushed by the Iron Claw. Good movie too, by the way. Everybody should watch that. But anyway, we're not. This isn't a wrestling podcast. It's a science podcast. Maybe I'll do a wrestling podcast one day. I don't know. But yeah, the topic for this next couple shows is mRNA. So I know a little bit about it. So are you ready for me to razzle and dazzle you with the information I know?
Please enlighten me.
Okay, so you have DNA, right? That's dinonucleic acid. Is it dino?
Close.
Okay. Yeah, it's D-nucleic acid, something like that. The crap they used to clone the dinosaurs in Jurassic Park. Okay, so you have that. That's completely separate from RNA, and RNA is ribonucleic acid.
Yes.
Right, and that's in your cells. And then mRNA is like a messenger form of that. So it's basically like somebody creates a letter and gives it to this, we'll call it the mRNA guy, like the postman, and the postman takes it down to the cells and says, hey, these are the orders from this guy, this is what you're going to do, and the cells say, okay, I'm going to do that, and adjust themselves accordingly to what's in the letter. Am I close? Am I way off, or do
That's
I fail?
A pretty good analogy. Yeah, no, so, I mean, I'm a microbiologist, so I'm mostly thinking about how this works in bacteria and archaea, which is a little bit different than how it works in human cells. But basically, so you start with your DNA, which is deoxyribonucleic acid.
That was it. I said
you were very close.
I associate with dinosaurs as pretty much everybody does now.
Well, yeah, you were thinking of Jurassic Park, but
Yep.
it's cool. So you got Mr. DNA, right? So Mr. DNA has all of the instructions for making pretty much every single protein that your cells need. And it's the proteins that do all of the work in your cells. So they carry out chemical reactions. They form the structure of the cell. They act as transporters. gateways into and out of the cell, signaling molecules. So all of the work of the cell is done by protein, and DNA contains the instructions for making every single protein that makes up the organism, that does all of the different jobs.
Okay, so let's go back
So
to
our
proteins a little bit real quick because
Sure,
I
yeah,
think
yeah.
let's explain more on what that is because I know it's different from like I drink a protein shake every morning and I know I'm not putting a bunch of like programming proteins and that kind of stuff. Like I know it's stuff specifically to build a muscle, but I know the proteins that you're talking about are slightly different than what you have in your shake each morning.
Yeah, well, so like the protein that you need in your diet, you can then use to make other types of protein, for example, the proteins that make up your muscle cells, right? So, all proteins are made up of these very simple building blocks called amino acids. You might have heard of essential amino acids, ones that
Mm-hmm.
you have to get from your diet. If you look at the nutritional information on the back of a can of protein powder, it'll tell you the amino acid content of that protein powder, usually, which ones you're getting and at what percentages. And so those amino acids are used to make proteins of every different persuasion with all different types of jobs. So when you get protein from your diet, whether it's by eating meat or drinking a protein shake or getting it from beans and rice or, whatever your favorite mode of consuming protein is, your body breaks down that protein and then uses it to build other proteins to
Okay,
do different jobs.
so super simplifying it then, like because I'm a big Lego guy. I have a giant box full of Legos, just miscellaneous parts, right? Those are amino acids.
Yes,
Okay.
exactly.
Then if I take proteins, is that kind of like coming up with a plan to build those amino like the Legos into something, or like looking through a Lego instruction book and saying, all right, I have all these pieces, I'm going to use that to build it, or is the proteins going to be something else?
The protein is what you're building out of the Legos.
Okay, so it's like the little house I built or whatever.
Right, the DNA is the instructions to tell you how to put those Legos together to make whatever it is you want to make.
Gotcha. All right, so it's the 8,000 page book I have on how to build the Millennium Falcon. So
Right,
that's
correct.
the DNA. The Millennium Falcon is the protein, and the amino acids are the 6 billion parts that I spent about $800 on.
Right. Except in your cells, there's only 20 different types of Legos, and that is it.
Oof, that I couldn't.
Yeah. Yeah. There's only 20 different Legos, and every single protein with any kind of function is made up of just different assortments of those 20 different Legos.
So, and how many like do we know how many I guess different builds you can make out of that assortment or?
I mean, it's theoretically like infinite because proteins can be different sizes. They can be small, they can be big, they could be made up of multiple smaller subunits.
Got it.
Some proteins are incredibly complex and others are really simple. It depends on their job and what their purpose is.
Okay, so you have the pieces, but they can be like stretched and shortened, like basically at will.
No, I mean the pieces don't change, but
Okay.
like say you could have one thing that you're building out of 100 Legos and another one that's built out of 1,000.
Oh, I
Or
gotcha. Okay.
you build up one section and then you build up a separate section that's shaped differently. You put those two together and now you have a functional whole. Stuff like that.
Okay, so you had those 20 pieces, and eventually, and I guess you could do that with real Legos too. I mean, eventually you could take those 20 pieces, and if size isn't an option and the number of pieces you have isn't, like, you could arrange those into infinite things. So, okay, cool. So
Yeah.
you have an
Yeah.
infinite number of things you can build out of 20 Lego pieces. Got it.
So the DNA is the instructions on how to put those pieces together.
Okay.
You probably know that the DNA, the genetic code, is 4 letters,
Mhm.
right? A, C, T, and G. So those are what are called nucleotide bases. And the order of those bases tells you what order to put the amino acids in when you're building a protein.
So that's like step 1, step 2, step 3 of the Lego
Right.
instruction book. Okay.
Right. So the code, the genetic code, is essentially made up of words that are 3 letters long. And these words are called codons. So every codon is 3 nucleotide bases, A, C, T, or G in some order. And those 3 bases call for a particular amino acid.
Okay, so when they do like the bigger Lego sets, they split them into bags. So you'll have like, say 10 different bags. So I guess we can call each codon a bag because when you look at the Lego instruction book, it's separated like steps 1 to 5 is bag 1, step 6 to 10 is bag 2, and so on and so forth.
It's brick by brick, really.
Okay.
So it's
okay.
like, yeah, it's not even like that broken down. It's just a string of this amino acid, then this one, then this one, then this one, then this one, then this one in a chain.
Got it. Okay. So it's not so much like bags, it really is just like one instruction per piece.
Yes.
Okay.
Yes.
Cool.
And then that long chain of amino acids that is made as the protein is being built folds on itself in a particular way to make the 3-dimensional structure of the protein. And that's driven by which amino acids are near each other, how they chemically interact with each other. Sometimes you have other proteins whose whole job is to help the protein fold correctly. They're called chaperones. So it's a whole complicated process to take this code that's in your DNA and then turn it into a functional product with a job.
Okay, so I've heard the term folding before, and I was always like wondering, is that literally like if I take a piece of paper and fold it in half, that's one job. If I fold it in half again, it's a second, like, I guess, job. Or is that kind of like it?
I think it's more like it's like if you had a really long piece of string, and in some places it was coiled, and in other places it folded back on itself, and in other places it was all just kind of bound up together. Some places on the string become physically connected to each other.
Okay,
but all of those different interactions have to happen before the protein will actually work properly.
Okay, so it's kind of like taking just like a big, like a fishing line and dropping it on the floor. But in that case, it's random. But in this case, it's actually organized and all for a purpose.
Yes, it's all directed.
But they kind of wind up looking the same.
Yeah, I mean, if you look at like the X-ray crystal structure of a protein, it can just kind of look like a big glob, but it can also have parts that look very ordered and structured. Things called alpha helices, which sort of look like a telephone cord. We're showing our age, right? Mm-hmm. Telephones don't even have cords anymore. But like the coiling in a telephone cord, or there's other structures called beta pleated sheets that kind of where the string folds back on itself multiple times in a zigzag, and it makes like a big flat piece as a result. So yeah, there's portions that look more ordered and structured, and then there's others that look a little bit more like globby and random.
Okay, so timey-wimey, wibbly-wobbly stuff. Got it.
Well, basically, but the way that the protein folds on itself is sort of embedded within the amino acid sequence. So like the way that the amino acids are lined up directs how this whole thing is supposed to fold on itself and become something that actually functions properly.
Okay, so it's like putting piece by piece together, and it goes off in however many directions or whatever connects to whatever, and then it becomes something functional.
Right, and then if you have it misfold, for example, into a hyperstable structure, you get a prion, and then you get mad cow disease.
Oh, lovely.
Yeah.
I don't think I've ever gotten mad cow disease from Legos, although, I mean, I have ordered some, I will confess, I've ordered some knockoff Legos, and those may or may not have been just basing by the way they smelled. I may have released something unholy into the world, I don't know, but they came from interesting places. So, who knows, maybe that is one of them.
Do the knockoff Legos work with the regular Legos, or nah, you can't mix them together?
No, they do. They do. Sometimes they'll fit a little bit looser. Sometimes they'll fit a little bit tighter. But yeah, they do a pretty weirdly good job of making these knockoff Legos. But can't be
apart from the strange odor.
Yeah, apart from the strange odor, and that's probably because of like what they use for the plastic. Because I mean, Lego uses like quality and all that kind of stuff. But whoever's making these knockoffs, I'm sure, are using like the El Cheapo plastic in some crazy factory in God only knows where.
Oh, some carcinogen.
Yeah,
somewhere, probably.
Totally some carcinogen.
Okay, so we have DNA, we have protein, RNA is sort of the middle messenger between those two things.
Okay.
So, in human cells, for example, human cells have a nucleus, which is basically like this compartment in the middle of the cell where the DNA is kept. It's like you have a vault where you keep all of the instructions, all the top secret information for how to make every single thing that you need for the cell to function. To make the proteins, you need another piece of cellular machinery called ribosomes. So ribosomes are like these little balls of protein mixed with a special type of RNA, and their whole job is to take instructions and make proteins. So they're like the factory manufacturers. They're like the guys working in the factory line.
Okay.
In order to get the instructions to the ribosomes, to your factory workers in your assembly line, you have to give them a set of instructions. And rather than give them the entire set of instructions for making every single protein, you're going to give them the instructions for the thing that you need made at that particular moment.
So kind of like an assembly line situation?
Yeah.
Okay.
Yeah. So there's some proteins in the cell that your cells need all of the time. So those genes are always what we call they're turned on. They're always activated. They're always being used to make something. Then other times there's certain proteins that you don't need all the time. You only need them under certain circumstances. So those genes are turned on or turned off. Additionally, I mentioned that like you have the entire set of instructions to make every type of cell in the nucleus of all of your cells, right? So your entire genome is contained in the nucleus in your brain cells, your stomach cells, your muscle cells, your, wherever. But those cells don't want to express the wrong genes and become the wrong cell type.
All right. So hypothetically,
right?
like a brain cell could become a stomach cell. It just doesn't want to.
Those genes for becoming a stomach cell will never be turned on in a brain cell because that would be really bad.
Right. So you have like, all right. So I'll equate it again to the factory line. So you're building cars, right? And you can build like there's one car manufacturer. You can build whatever the heck car you want. You can build Fords. You can build Toyotas off this assembly line, and everybody builds a wheel because you need a wheel in all cars. But some will build like the Mustang body, some will build the Prius body, and some will build like the F-150 body.
Yeah, essentially. Those decisions are kind of made very, very early on in development. So when you're an embryo, certain cells decide to become your bones and other cells decide to become your brain. And so stem cells are cells where those decisions haven't been made yet, so any gene can be turned on. But you do get to a point in your development where those genes for other cell types can never get turned on.
So they're freshmen in college before they choose their major.
Yeah, it's kind of like that.
Okay.
But then, by the time you are a fully formed fetus, your muscle cells are never going to become bone cells or stomach cells. They've made their decision. They're seniors. They're about to graduate. They're not changing their major.
Yeah, you can't they can't switch majors 10 times and wind up as a communications major and ultimately regret their life's decision. Me.
Emotional damage.
Ouch.
That felt a little close to home there.
A little bit. That hit. I hurt my own feelings.
I'm sorry. So the mRNA, messenger RNA, is a type of RNA that's used as a set of instructions for the ribosomes to make proteins. So it's like you've got this vault containing the information on how to make every single type of protein. When you want to make a particular one, you make a copy of the instructions to make that particular protein, and then it gets sent out of the nucleus and it's given to the ribosomes so they can start making that protein.
Okay, so somebody goes to a copier and copies the instructions, sends it off to this postman guy. Who's deciding who gets what instructions?
Oh, so...
Or is that like a whole other topic?
Well, any ribosome can make any protein. So
Okay.
pretty much once the gene has been what we call transcribed so copied into mRNA, and it gets sent out of the nucleus, any ribosome can grab it and just start making protein from it.
Okay, so it's like I guess it's kind of like an Uber situation. Well, no, Uber assigns it, but Instacart, because they all pop like the jobs up on the screen. You pick what you want. So it's a situation like that. Okay, cool.
Yeah. So a ribosome encounters a set of instructions, and it's like, all right, I'll start making this. Cool. And that's going to come up later when we talk about how mRNA vaccines work.
Yeah, because I was going to go be even bigger and be like, all right, who decides on the instructions? Like, who is Instacart versus the workers just picking? But yeah, if we'll talk about that later, I'll save that one.
Well, so it's a really complicated system that determines what genes are copied and when. And there's loads of different mechanisms and layers and layers and layers of control over what genes are turned on and what genes are turned off. And some genes are under multiple layers of control, and it can happen at different points along the process. So it's a whole complicated thing that I'm trying to make as simple as possible.
Oh, fair enough. All right, so that'll be like a whole other thing. That's a whole nother thing.
That would.
Okay.
Yeah, gene expression would be a whole other conversation. But the way that I explain it to my students sometimes is like, if I had a cookbook and I wanted to give someone the recipe for something, I would just give them a copy of the one recipe and not the entire cookbook.
Right.
But there's an extra layer onto it, and that is that the language that the messenger RNA is in is slightly different than the language of DNA. So you're still using this 4-nucleotide base system, except the letter T is replaced with the letter U. And RNA, unlike DNA, is usually single-stranded. So you know how you look at a picture of DNA, it's got that double helix shape, that classic Watson and Crick shape, Rosalind Franklin shape. The
Google
two-strand.
Mr. DNA. Just make it easy. Yeah, Google Mr. DNA from Jurassic Park. Yep.
Curled around, like coiled around each other like a twisted ladder, right? Mm-hmm. So RNA doesn't look like that. It's just one strand. It's like a ladder, but with only one side to it.
So a stick, basically.
Yeah, like a stick with like little rungs sticking out on one side.
Yep.
Right. So imagine if I had a cookbook and it was in Italian, and I wanted to give someone a recipe. They don't speak Italian. I have to transcribe the recipe into English.
Yeah, I place that blame on the person. Download Duolingo and learn yourself some Italian.
Yeah, well, we should all be bettering ourselves with
yeah,
learning new languages, right? I'm working on French right now.
But... I'm working on Japanese, so that's
right on.
Yeah, that's a lot of fun. I want to pull my hair out with the written language part of it, but it's getting there.
I can't imagine. At least in French, I'm using the same alphabet.
Yeah.
And a lot of the words look the same, so it feels a little bit like cheating.
Yeah, now all the Latin-based languages, you can kind of figure it out, but Japanese is a whole other, and that's always been one interesting study I've wanted to try to. A topic I've wanted to get more into is like, how did we get? Because if you look, if you go by, I guess, biblical history and whatever histories you go all the way back to everybody spoke one language, and then all of a sudden they had the Tower of Babel, and that's where everybody got scattered around to speak the different languages of the world. So how did we get from like that one unified language to an offshoot of the Latin, Germanic-based languages, and then you get the Asiatic languages that sound way completely different? Like that's what I've always been interested in too, is how those split apart. I mean, I guess that counts as science, so maybe that's a topic we could maybe explore later. But
there's a whole like study of linguistics of like how languages have evolved and which languages evolved from which ones and which ones are more closely related. It's almost like an evolutionary tree, right? But for human language, which is really fascinating.
Yeah. So coming soon to an episode near you, the science of linguistics, but.
We'll have to find somebody who knows a lot more about that than I do.
Yeah, because there's a reason I haven't learned it yet. I haven't had the time. Anyway, back to the topic at hand.
So back to mRNA, yeah. So we make a transcription of the gene for whatever protein we want to make, Then we send that transcript out of the vault, the nucleus, and give it over to any ribosome who's there to pick it up. And they start turning that transcript into protein. And the same transcript can be used to make more than one protein. You can read those instructions over and over and over again and make lots and lots of protein from the same transcript. But eventually, the transcript self-destructs. It does not last forever.
Oh, Mission Impossible stuff right there.
It's sort of like, did you ever watch Inspector Gadget as a kid?
Yeah.
You remember how he would always get a little slip of paper with his mission for that episode, and at the end it would say, this message will self-destruct, and then he'd like throw it away and it would explode.
Mm-hmm.
It's kind of like that.
They stole that from Maxwell Smart.
Yeah. So this is really important, actually, because it means that if a gene is turned on, if you're making protein from that gene, it's not turned on forever. Once the mRNA is made, it lasts for however long, and then it destroys itself. So you have to keep making transcripts of genes that need to be on all the time. But if a gene needs to be turned off, that mRNA eventually destroys itself. So if you stop making transcripts of that gene, eventually you'll also stop making proteins of that gene because the transcripts will go away. They disintegrate.
Okay, and so on a bigger scale, what's the effect of that? Is that like how you stop growing, or is that like, what's
It's part of how your cell controls what proteins it's making and when.
Okay.
So that if there are proteins that you need at particular times, but not others, you can stop making that protein because you stop making the instructions to make it.
So if you're like, all right, so if you're like a pro athlete, right, and you're always building muscle, and then you retire, you don't need to build as muscle as much. They turn off. Is that?
Well, I get it's probably a bit more complicated.
Yeah, I know. I'm oversimplifying it, but.
Well, so there's like certain genes that are turned on, during your development.
Okay.
In the womb.
Got
right?
it.
That are turned on in order for you to develop into what we recognize as a human shape, right? With 2 eyes and fingers and toes and all of that stuff.
Oh, okay.
Those genes are turned off after you're done developing.
Okay.
And they're never turned on again. So, like, those transcripts to make the proteins needed for those processes, they don't hang around. They're eventually destroyed because you don't need those genes anymore.
Yeah, and if for some reason they kept like staying on, you would wind up looking like a biblically accurate angel or something like that.
Right, or something out of like John Carpenter's The Thing.
Yeah. So then that, I mean, I don't know if you can
Cronenbergian monster.
Eldritch horror.
Eldritch horror, exactly.
Then I don't.
Shoggoth.
Yeah. Do not say their names. My goodness, I'm looking at some woods. Do not say their names. Then I guess the natural question to ask about that is, like, are we working on ways to turn that, like, if you get your finger cut off, obviously we don't have the things anymore to regrow a new finger, but is there any way that, like, are they working on ways, or if they just kind of shut this out completely as a non-possibility, but have they said maybe there is a way that we could temporarily turn those back on and regrow a finger?
I don't know. Actually, if there's a way, I mean, I presume that there are ways to do that. So we're looking at ways that we can edit the human genome using something called CRISPR-Cas9, which is a system that bacteria use to defend themselves against viruses. And to put it pretty simply, it's basically like the bacterium keeps a little bit of the viral genome so that it can recognize that virus if it gets attacked by it again. And so we are kind of leveraging that to be able to deal with genetic disorders. There's lots of different genetic disorders, congenital disorders, that are caused by a single mutation in a gene that causes the protein that's made from that gene to not function properly. And when that happens, it causes these debilitating illnesses. So things like sickle cell anemia or Tay-Sachs or cystic fibrosis, these are all caused by single mutations in a single protein.
So
So
that
if we
would
could go
be
in,
so that would be like the protein was supposed to fold right, but it folded left.
right.
Okay.
So there's one amino acid is switched out for another one somewhere in the code, which causes the entire protein to fold wrong, and now it doesn't work.
Yeah. I've done that many times with Legos. Makes perfect sense there.
Exactly.
Yeah.
Exactly. So if you can go in and fix that error, so now it makes a functioning protein, you cure the disease.
Hmm.
So it's possible you could edit the proteins that control what genes are turned on and off as well, and encourage certain genes to be transcribed that aren't normally transcribed. I don't know what the implications of that would be or how easy it would be to control is the thing.
Yeah. Yeah.
Now we're getting outside of my understanding, into like human anatomy and physiology, which is not really my wheelhouse. I enjoy it on an aesthetic level, but not so much a scientific one.
So I guess that's probably then what they're trying to figure out. Like, I mean, obviously they've studied the DNA and genes and stuff of lizards and other creatures that can regrow limbs and stuff. So I'm guessing then maybe they have the know-how. They just don't know how to limit it and stop it. And even worse, stop it from falling into the wrong hands. So we have a bunch of guys running around like the Incredible Hulk.
Yeah, there's all sorts of like ethical implications to genome editing. So like, yes, it would be very cool if we could end these debilitating congenital diseases, but then what stops somebody from editing their genes to make designer babies? And is that okay?
Mm-hmm.
What are the ethical implications of, the ableism wrapped up in that?
Yeah.
Trying to create the perfect baby. It's a little cringe, right? So there's a lot there's a lot there that we could talk about, and I feel like it could be a whole other conversation.
Yeah, we could do an ethics of science, a huge
Absolutely.
thing. Jeez.
Oh my gosh, yeah, we could do so many different episodes about ethics of science, ethics in science. It's a whole topic.
That I know a little bit about, of like the human experimentations and stuff like that. Oof. Ugh.
Oh,
I don't want to think about it.
Yeah.
Anyway, moving on.
There's a lot of ugliness there, but that's a topic for another time. So yeah, I mean, the bottom line is that all human cells contain mRNA at all times, and that mRNA directs your cells to make the things that it needs to make in order to function. And it's a really cool process. The whole process of going from DNA to RNA to protein is what Francis Crick, one of the co-discoverers of the DNA structure, called the central dogma of molecular biology. He kind of meant it as like a tongue-in-cheek kind of thing. Like, it's not really a dogma. Like, there's more to it than that. But it's sort of like the central pathway that drives information flow in your cells. You've got the vault containing the information to make every single thing you could possibly need. Then you've got the mRNA, which acts as your set of instructions for the specific thing that you need. And then the protein, which is the final product, which does whatever job you need it to do.
Okay. That makes sense. Like, surprisingly, it makes a lot of sense. So they had the jobs posted. You have a courier come get it. You have the courier pass them off to the proteins. The proteins say amino acids, you're building it this way and that way, and then the amino acids create the fold proteins, right? And Bob's your uncle.
Yeah. You put all of these different amino acids together, and you create any number of different amazing things. And like, the amount of diversity out there in terms of how many different proteins there are and how many different things that they can do is just mind-boggling. Like, there's so many that we don't even know what their function is. When we sequence genomes, we find a whole bunch of information that looks like it probably codes for a protein, but we have no clue what that protein does.
Gotcha. Cool. Well, that sounds like a kind of almost like a perfect stopping point for today's episode, because then I guess next time we can talk about some applications and some, I guess, more technology and cool stuff that's coming along with it. So yeah, thank you for that. That was Yeah, that was. I thought it was going to be like this crazy complex thing, but you broke it down very nicely and very simply, so even I could
That's the
understand it.
deal.
That is what you do. All right,
Yeah.
so the big question is, as we mentioned, we want to start setting up, I guess, playlists, and if I can figure out how to do it on Spotify, which should be simple enough, we could post them.
Hmm.
But have you given any thought to a couple songs that would go along with mRNA? Maybe like two or three of them that we could throw down there.
Yeah, well, I think we talked about "Gotta Get a Message to You"
Yep.
by the Bee Gees,
I was going to say, are you
or the
going?
Moxie Fruitus
Yeah,
cover.
are you going Bee Gees or Fruvis?
I like the Moxie Fruitus cover, but only the live a cappella version. I don't like the album version.
Fair enough.
I know that's so specific.
Moxie Fruvis is
Message
so
in a
specific. There's going to
Bottle.
be like two people out there who know who they are.
I was like, who the hell are they? Um, Message in a Bottle.
Yep.
Is that the Police or is it Sting? I don't remember.
Sure. I think it was the Police.
Okay.
I don't know. I don't. I'll
Maybe
look it up later.
Police. I forget.
Somebody's going to add
Yeah.
us in the comments about that. Like, come on, man.
Yeah.
It was
Look,
released in
I know
1975 when he was with the Police, but then 2 months later he became Sting, but then he became the Police again.
I know two things about Sting. I know that he's into tantric sex, and he was in a tiny little bikini in 1984 Dune as Fade, and he was amazing in that part, and not just because of the tiny bikini. He's actually a really good actor.
I used to get him confused with the wrestler Sting when I was a kid. No surprises there. I was thinking.
Here we are again, back in wrestling. It's like
I know. I really need to do
Sounds
that. I've been talking for
familiar.
years about doing a wrestling podcast, and I really should. Um, yeah, I was thinking, let's go with some Joko, Code Monkey. Just classic.
Oh, yeah.
Mm-hmm.
Actually, yeah.
We'll
Yeah.
go Code
That's
Monkey, and then I was struggling with the other one because there's so many. There's a song by Starship, and it has message in the title. I can't think of it now. I looked it up earlier, but got to get some got to get some Starship on the playlist. I'll give you an IOU on that one actually because Code Monkey
fair
was the first
enough.
one.
Fair enough.
Yeah, I could sit here and take some time to think about a really good one, but because there's a ton of songs about like sending messages and like and letters and like hey Mr. Postman could be an
I
obvious
was
one.
just thinking of that one. Like you took it right out of my brain.
Yeah,
Mr.
and
Postman.
that is a great song, but I mean I want to go something more, you know. Although I guess if we're going like a wide variety of things to listen to, that covers it. Because we got some
I would say
Motown, we got
so.
some Canadian folk, we got regular rock, and we got whatever Joko is. Nerd tunes. So
Yeah, I think that's a pretty good place to start.
yeah, we'll get a couple ones up there on the playlist, and you know if I ever get it created. But yeah, I think that's gonna do it for the episode. Shower guy, you got any thoughts?
Well, all this talk about proteins and amino acids got me kind of hungry and I wanted to go out, but I'm a little bit worried because do these jeans make me look fat? Stay cool and stay clean.
Cool. Well, thanks, Shower Guy. And Lauren, thank you very much for hanging out today. And
My
don't
pleasure.
forget to like and comment and more importantly, well, most importantly, subscribe to the podcast here because we're growing an audience. We want to get your guys' participation. Let us know some of the topics you might want to hear about. Really, anything that has to do with science, we'll give it a crack maybe in a later episode, possibly much later on down the road because not only is it about exploring Lauren's expertise, but it's also about learning different things. So we could always learn some other topics together. But that is going to do it for today. So thank you guys all for listening, and we will see you
Thank
next
you.
time. Yeah. Say goodnight, Lauren.
Yeah. Good night, Lauren.